Welcome to the first of the two screencasts on cloning. This first screencast distinguishes between two types of cloning and then goes on to look specifically at cloning in plants. Okay, before we start looking at this aspect of cloning, one thing to distinguish is the idea of gene cloning and cell cloning. This particular screencast looks at cell cloning, whereas we looked at some techniques in previous screencasts on genetic engineering on processes which would really be gene cloning. So PCR would be an example of gene cloning, as would putting plasmids with new genes into bacteria and allowing them to reproduce and multiply would also be an example of gene cloning. Okay, but these particular screencasts are looking at cloning of cells and of whole organisms. Okay, so dealing with this, first of all, there's two distinct types of cloning which we're interested in when we're looking at cloning of cells: reproductive cloning and non-reproductive cloning. And this non-reproductive cloning is often referred to as therapeutic cloning. I'll come back to that in a second. Reproductive cloning, this is any process that generates identically new organisms. Okay, so this happens naturally, obviously, in the production of identical twins, or monozygotic twins as they're sometimes known. But we can do this by a number of ways. We can split embryos, we can do nuclear transfer, which we'll look at a little bit later, micropropagation of plants, which you look on this screencast, and artificial propagation or vegetative propagation in plants, which we'll also look at. Okay, so reproductive cloning is actually producing whole new individuals, be they plants or animals. Non-reproductive cloning is the use of clone cells to generate tissues and organs, and this potentially has uses in replacing damaged organs and tissues in various diseases or accidents. Okay, and I've got some advantages here. So the cells that you use, okay, for transplanting or whatever, would be identical, therefore there wouldn't be any immune response and tissue rejection. Potentially there's a very large potential amount of donor organs that could be produced, or donor tissues that could be produced, and therefore the issues that we talked about with xenotransplantation would be avoided. Cloned totipotent cells can differentiate into any type of cell. Okay, so totipotent is a word that we learned in F215. A totipotent cell is a cell that can differentiate into any type of cell. Okay, and also the use of cells, putting cells into structures to replace damaged or worn out structures, avoids invasive surgery. Okay, and the type of things that might be able to happen in the future is regeneration of heart tissue by putting cloned cells into heart, and they would develop into heart tissue to replace damaged heart tissue. Repair of nervous system following diseases, and also perhaps repairing spinal cord after damage is done to it. Okay, anyway, a little diagram down here just to outline the differences between cloning to produce individuals and cloning to produce cells. Okay, so here's typically how we might produce a cell. Okay, so we've got a somatic cell which has got a nucleus of the organism we're trying to clone. We've got an egg cell of the same species. We remove the egg cell to create what's called an anucleated egg cell. We then insert the nucleus from the cell we're trying to clone into our anucleated egg cell, and that produces a ball of cells, okay, which could be called a clone. Okay, now what we do with this clone, okay, is we'll determine whether we are doing reproductive cloning or non-reproductive cloning. If it's reproductive cloning, we would take that ball of cells and we could implant that into the uterus of a surrogate mother, and that ball of cells would develop into a new individual. Okay, in terms of plants, this ball of cells, which we call a callus, okay, a callus could be planted into particular nutrient mediums with the right growth hormones, which would develop shoots and roots, and that would develop into a new individual plant. Okay. In terms of therapeutic cloning or non-reproductive cloning, we produce this ball of cells, which might form an embryo, which we then can use those cells. We could use those cells for various aspects of the use of cells for regenerative tissue transplants, etc. Okay, so that would be therapeutic or non-reproductive cloning. So reproductive cloning for individuals, non-reproductive cloning, production of cells for transplant, etc. Moving on to the next slide. This slide requires us to be able to describe the production of natural clones in plants, okay, using the example of vegetative propagation in elm trees. Okay, so in many organisms, cloning occurs in forms of asexual reproduction. Okay, we'd have looked at this before at GCSE, things like strawberry plants producing runners, potatoes, things called tubers, which are swelling in roots of potato plants, which then the following season will sprout and develop new potato plants. Okay, the example that we're required to know here involves structures called basal sprouts on roots, or root suckers as they're more often known as, a process known as suckering, and this occurs in elm trees, okay, which used to be a very large part of the English countryside. There used to be elm trees everywhere, not so many now for reasons I'll explain in a minute. But anyway, they have a mechanism of asexual reproduction where roots that run close to the surface of the ground, what they do is that they throw up sprouts, basal sprouts, which grow vertically upwards. Okay, and what happens is that can lead to the production of a new tree. Okay, and the suckers will eventually take over from the parent tree. Okay, so showing that diagrammatically, here is a tree of some sort. Okay, and here's a root, a shallow root, and here is a sucker growing up from that root. Okay, so it's similar-ish to the runners, which grew from strawberry plants, but runners were horizontal stems that ran across the ground and then hit the ground, and then roots grew from that. These are actually roots, okay, growing horizontally underneath the soil, which then throw shoots up from the roots to form new plants. Okay, in terms of the elm trees, okay, the advantage of this for elm trees. So if you can imagine, here we have woodland, and these slightly darker green trees are elm trees, and these lighter green trees are something else. Okay, now what can happen is that these suckers can grow up here, obviously not hugely useful, because obviously they're in the shade of their parent plant, and therefore they won't grow particularly well. Okay, however, if the plants are attacked by some sort of it could be a forest fire, it could be they could be cut down, or it could be over foraged by animals, or whatever it is. Okay, so the plants have been removed. Okay, now our elm tree suckers are already partially developed. Okay, and they will quickly colonise the area. Okay, to the detriment of the competing trees. Okay, so the elms would very quickly colonise the area, and you would get a cluster of elm trees. Okay. Now the problem with this is that, of course, all these elm trees here, this elm tree here, and this elm tree here, and this elm tree here, are all clones of the original parent elm tree. Okay, then we have a very limited gene pool. Okay, so they're genetically all identical to each other, all of these plants. Okay, and there are huge disadvantages of these, as was demonstrated by the elm tree. The reason I mentioned elm trees used to be a key part of the English countryside, which they no longer are, due to the fact there was a disease called Dutch elm disease. And obviously the Dutch elm disease, which was carried by beetles, affected elm trees, okay, and the elm trees died, as we can see here, okay, but very quickly you can see that the suckers, okay, which are genetically identical to parents, would also start to develop this Dutch elm disease too. Okay, so the reason that might be is that if we had a properly sexually reproducing population of elm trees, there would be sufficient genetic diversity amongst those elm trees that we would probably find elm trees which were resistant to Dutch elm disease. But because they reproduce asexually in this way, well, the majority of these trees are produced by asexual reproduction, or vegetative propagation as it's known, is that they're genetically identical, and therefore if the parent plant succumb to Dutch elm disease, the likelihood is that the offspring would do too. Okay, so this has had a catastrophic effect on elm trees right across Europe and large tracts of America too. So in summary, many plants have mechanisms of cloning or asexual reproductions. We talked about runners and potato tubers and so on. When sexual reproduction occurs via growth of non-reproductive organs such as stems and roots, it is known as natural vegetative propagation. Okay, so that's a key idea you should know about natural vegetative propagation. Okay, which is things like suckers, tubers, runners, etc. Natural vegetative propagation. Okay, the example you've got to know about is elm trees. Okay, and they use things called suckers, or they're sometimes known as basal sprouts. Okay, and they grow from stem cells. Remember, plant stem cells are called meristem cells. That's an F215. And these meristem cells can be found in shallow roots. Okay, and as a result, these shoots grow up from these roots. Okay, and when parent tree dies or is under stress, they can grow and replace the parent tree. Okay, and when they grow, they eventually grow into an individual tree in their own right. Okay, advantage: root suckers ensure patch of ground is occupied by elm trees even once parent tree has died out, has been coppiced or burnt down. Disadvantage: parent trees succumb to a disease such as Dutch elm disease. It means all the trees and all the suckers will succumb to the same disease. Okay, so quickly moving on to the next. This slide looks at artificial clones. Okay, so vegetative propagation, okay, is the natural formation of clones such as suckers and runners and tubers that we talked about, but man has very quickly discovered that we can do this artificially, and this process is known as artificial vegetative propagation. Okay, and we've been doing this for many years. Okay, for those of you who are interested in gardening, you'll be aware of the process of cuttings. Okay, we take a stem from a parent plant, we usually cut it just below the node where we know there's going to be a node being somewhere where a new shoot is about to grow from, or a new branch is about to grow from, where we know there's going to be meristem cells. Okay, we treat the cut end with hormones called rooting powders, and then we can, after roots have started to grow, we can then plant our cutting, and the idea is it will grow into an exact clone of the plant you cut it from. Okay, so a lot of gardeners will do this to avoid the expense of having to buy new plants from very expensive garden centres. Grafting is another thing that can be done, which is when we can join two plants. Plants don't have the immune system issues that animals have, and therefore we can actually graft on bits of plants to other plants. Okay, so what we do is we cut the stem of a recipient plant in such a way that the stem of another plant can be inserted. Okay, we have to align the vascular bundles, so water will move up the xylem and sugars will move down the phloem, and what will happen is the graft will start to grow. Okay, so we can graft pear trees onto apple trees, and all sorts of interesting things can be done with grafting. I'll show diagrammatically below in a second, and then micropropagation, which we'll also come to. So dealing with the cuttings and graftings first of all. Here's a little diagram to illustrate cutting. So here we've got a plant of some sort, and we've cut it. Tend to cut them just below where there's been growth, a lateral growth, so that we know that there's going to be meristem cells here. And then what we do is that we insert these cut ends into some form of rooting powder, which is an auxin, which will promote the growth of roots. Okay, so roots will start to grow at the bottom. Okay, and then we can plant our cuttings. Okay, so we've planted this cutting into here and this cutting into here, and we've got two plants which would be identical to the original plant. Okay, that process is known as making a cutting. And moving on to grafting. Okay, so this is a lot of roses are produced in this way. Sometimes we find that we have a plant which have got some desirable characteristic, like a particular coloured flower, but their rootstock is quite weak. Okay, and then we might have another plant which has a good strong rootstock. Okay, but the flowers aren't particularly desirable. So what we can do is we can cut this plant here. Okay, with the less desirable flowers on it, okay, with the desirable rootstock. Okay, we can cut off the top of that plant and insert, okay, being careful to line up xylem and phloem, insert this plant into the cut end, this bit in the stock. Okay, and we insert that in there. Okay, and as a result, these healthy roots, okay, will provide lots and lots of water and nutrients to support this plant here. As I say, a lot of roses are cultured in this way. Okay. So moving back to the slide, the next thing we had to look at was the idea of micropropagation, okay, or plant tissue culture, which is a more modern technique, okay, which enables horticulturists to produce vast quantities of clones of a particular plant with particularly desired characteristics. Okay, and the way it's taken is that we find what we call apical meristems. So the tips of roots and tips of shoots are good for this. Okay, and we cut those from a plant, and then they become known as X-plants. Okay, and then the X-plant is then put onto a growth medium. Okay, which produces a ball of cells called a callus. Okay, and these cells are all undifferentiated cells. Okay, because they originate from these stem cells or meristem cells. Okay, forms a ball of cells called a callus. Okay, we can then separate the cells from our callus and place those single cells in growth medium. Okay, and the first thing we do is we put it into a growth medium that stimulates shoot growth. Okay, so the differentiation of our callus cells into shoot cells. Okay, once the shoots start to grow, we then transfer our shoot into another growth medium which stimulates root growth. Okay, so we end up with our callus cells differentiating into shoots and roots. Okay, once they've grown to a certain size and they start to grow leaves. Okay, so they can photosynthesize, they can be transferred and grown in a glass house until they're large enough to be able to plant in soil. Okay, so again, seeing all that in a diagram below here. Okay, so here I have cells, so I tend to use, I've just used leaf cells here, but we tend to use meristematic cells of some sort. Okay, we put that into a culture which promotes the growth of a callus, which is a ball of undifferentiated plant cells. We can separate those cells and insert those cells into growth mediums. One medium which has a hormone which stimulates shoot growth, so the shoot starts to grow. Okay, and then we transfer them to another growth medium. This one develops roots. Okay, to prevent root growth. Okay, and once the growth it's a certain size, we can then transfer it to a glass house, let it continue to grow, and then it can eventually be transferred and planted into soil. Okay, and this plant here is an exact clone, a genetic clone of this plant here. Okay. So hopefully that illustrates that slide. Moving on to the plant hormone slides. The other thing you've got to know about is the advantages and disadvantages of plant cloning used in agriculture. Okay, so I've just listed 2 or 3 of these. I'm sure your textbooks might even have further ones, but here's a couple to keep you going. So the great advantage is the farmer knows the features of the crop before it grows. Okay, what its yield is likely to be, what the taste of whatever it is he's growing is likely to be, colour, level of disease resistance. All of these are known. Okay, and that can make his product more marketable, and things like disease resistance is going to make it easier to grow. Okay, it also will grow at constant rate. Okay, and therefore harvesting costs will be cut down if all your crop harvest becomes ripe at the same time. You only have to harvest at one time of the year, and that reduces harvesting costs. Okay, and then this idea of micropropagation. Okay, very quickly leads to huge numbers of clones being produced. Okay, disadvantages. Okay, the genetic uniformity, which up here is such an advantage, can also create problems. Okay, so it would lead your plants to be equally susceptible to a particular disease that might come along, and the widespread use of cloned plants reduces the gene pool. Okay, and this will prevent or reduce the possibility of developing new strains with different characteristics in the future. We talked a bit about this back in F212 when we talked about conservation and seed banks to try and conserve old and less popular strains of various plants, so that breeding programs can be done in the future to be able to develop new strains for new crops which might be required as the environment changes. Anyway, that concludes the first screencast on cloning, which looked at the basic idea of cloning. Reproductive and non-reproductive, or sometimes therapeutic cloning, was defined, and then we looked at aspects of natural vegetative propagation and artificial vegetative propagation, and looked at some of the advantages and disadvantages of those processes.